Hello. So very good afternoon to the student who have joined. Can you please let me know in the chat box if my voice is audible, and also if you can see the presentation I'm showing here. Okay, thank you so much for the confirmation. We will wait till more students join, or till 6 p.m. Then after that we will start our classes. Very good evening. I will wait 5 more minutes till more students join, and then I will start today's session. Meanwhile, I hope my voice is audible and the video is visible to you. If not, you can just let me know in the chat box if there is any problem with my voice or audio. Just let me know in the chat box. Okay, so it is 6 p.m. So I think I will just start with the introductions. Okay. So let me start with the introductions first. Okay. Yeah. So very good evening to all of you. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry. So today it is our week 5 live interaction session. So every week we are going with a week ahead with our live sessions. So today it is the week 5 live session and here we will discuss the week 6 course content. Okay, so last day it was in our week 4 session we had discussed the week 5 course content already. Right now the week 6 is online and you have to submit your assignments for week 6 soon. So today I will discuss topics and MCQ questions which are from the week 6 course content. So this I will discuss 20 MCQ questions and first you have to guess the answer, what is the correct answer for that particular question, and after some time I will show you what is the correct answer of that particular question and we will discuss that part. Okay, so for smooth streaming of the session I have just muted you all and kept your cameras off so that I can record the session without any problem, because I am recording the session. It will be problematic if any network disruption happens. So that's why I have kept your microphones off, but the chat box is open always. Just I have already said every day, like the chat box is open, and if you have any doubt, any query, you want to tell me anything about the class and any problem for previous classes also, just you can write it on the chat box. Okay, I am monitoring the chat box always, so you can just let me know any problem you are facing during the session or after the session. I will just share my email ID with you. So if you have any doubt, any query or any problem, just you can reach me through the email also. Okay. So before starting the session, just like always, I would summarize few points. So first, there will be this kind of problem solving session every week for 12 week. Plus we will have a summary session also at the end for 1 hour summary session, so that I can summarize all the whole course very well. Okay, and these sessions will be on every Tuesday from 6 p.m. to 8 p.m. and the sessions, it will be live on Google Meet platform. Okay, so this is the link for your class for every day. The link is same, the link you are using, the link is same for every class. So you can just save this link and use this to join the class every Tuesday. And if any network disruption happen from my side, so please wait for some time, do not leave the session. I will try to join as soon as possible. Okay, and during the session you can just post your doubts, your query, everything in the chat box. And if the network disruption happen from your side, you don't need to worry, because I will share this presentation and also I will share this video of lecture with you. Okay, and you can just go through that lecture materials after the class also. So if any network disturbance happens from your side, you don't have to worry. And if still any doubt persists after class, you can drop an email to me. I am always open for this email option. So you can just email me your problem, your queries. I will try to answer that query. And also there is a discussion forum on the SWAM website. Okay, for the course, this course medicinal chemistry, the code is noc24-cy17. So on the discussion forum also you can just write your queries about the assignments, the examinations and everything. You can just post your query and course member will take care of your doubts. Okay, and again I have just mentioned that this presentation and class recording will be shared to you soon. Okay, so as I have mentioned, today it is our week 5 live session where we will going to discuss the week 6 course content. So right now the week 6 is online. Okay, and we will discuss the topics which are in the week 6. So what are the topics in the week 6? Let us just check it out first. Yeah, so again, if you have any doubt, any query, please write in the chat box. Okay, whoever has raised the hand, just if you have any problem, you can just write it on chat box. I will just check your chat box and I will answer your query. Okay, so again I am saying, if you have any problem, just write it on the chat box. So I hope my voice is clear, right? And you can hear my voice and also you can see the presentation. If not, please let me know right now. I hope everything is fine with my voice and also the presentation. And if any problem is there, you can just let me know right now in the chat box. Okay, so what are the topics which are included in your week 6? So these are, first we have the pharmacokinetics. Okay, The pharmacokinetics is in the part 1 and part 2. Then we have drug metabolism, and this is this is divided into 4 parts, part 1, 2, 3 and 4, and then we will have a tutorial about ADME. So we will discuss about the ADME a little bit. Okay, so these are the topics for your week 6. So I think I will start the session. Meanwhile, when more students will join, they will also be part of the session, so we don't have to worry about it. Okay, so let me show you the first question of today's session. So how we do it, I will show you a question and you have to answer it in the chat box. Okay, even if your answer is wrong, it doesn't matter, but answering the question is very important. So I will hope that you will try to answer that question. First, read the question very carefully, read the option very carefully, and then write what is the option you think is the correct one, A, B or C or D. So the first question of today's session is, which of the following statements best describes pharmacokinetics? Your options are: number A, the study of how drugs reach their target in the body and how the levels of the drug in the blood are affected by various factors. Number B, the study how sorry. Number B, the study of how drugs can be designed using molecular modeling based on the drug's pharmacophores. Number C, the study of how a drug interacts with its target binding site at the molecular level. And number D, the study of which the functional groups are important in binding a drug to its target site and identification of a pharmacophore. So which of the following options, A, B, C or D, is correct one? Or which of the following options, A, B, C and D, best describes the pharmacokinetics, this word? I will give you some time, read the options carefully, and then let me know in the chat box which of the following you think is the correct one. Which of the following statements best describes pharmacokinetics? All of you should answer the questions. Even if your answer is wrong, it doesn't matter, but I want all of you to try to answer the question. Let me check the chat box now. Okay, so I am having few answers like number A and number A. Others also should try to answer the questions. Okay, even if your answer is wrong, it doesn't matter, because this is only for practice. So I want, for next time, I want all of you to answer the questions. Okay, even if your answer is wrong, but still you have to try. Okay, so I will show you now what is the correct answer. So yes, the correct answer is number A. Pharmacokinetics is the study of how drugs reach their target in the body and how the levels of a drug in the blood are affected by various factors. Now let us take a look what is the pharmacokinetics. So pharmacokinetics is the movement of a drug through the body from entrance to exit. So there are four factors basically for the pharmacokinetics. Okay, these are known as absorption, distribution, metabolism and excretion, which is abbreviated as ADME, A, D, M and E. Okay, so first point is absorption. So what is absorption? Absorption is the movement from the side of administration to bloodstream. Okay, common route of administration. So administration is like taking the first what is the first is the entrance of the drug in our body. Okay, so there are various ways through which a drug can be taken. Okay, the common routes of administration is the oral way, which is either in the tablet form or a capsule form. Okay, or in any kind which we swallow. Okay, that is the oral way. Then we have the intravenous or IV, means the which is direct injected into the veins. Okay, and then we have intramuscular, we have subcutaneous, we have transdermal, and we have pulmonary or nasal. So these are the common routes of administration. Then what is the most important factor in case of the absorption? The term is known as bioavailability, which is the extent and the rate at which the drug enters the systematic circulation. Bioavailability is 100% in the intravenous administration but varies widely from all other routes. Okay, so first thing is the bioavailability, means the extent to which this particular drug is taken in our body. Then we have the distribution. Distribution means the transportation through the systematic circulation to target tissues or organs. This distribution is influenced by adequate blood flow, ability of a drug to exit the vascular system, ability of a drug to enter the target cell. Okay, and the factors that may limit the distribution is the vascular damage due to the chronic disease such as diabetes, limited blood supply to the abscesses or the tumors, tight junctions of the blood-brain barrier. Then we have metabolism. What is metabolism? It is the chemical alteration or biotransformation of a drug to make it pharmacologically active or to facilitate the elimination. So first, after absorption and distribution, we have comes to the metabolism, where the drug is converted to the more absorbable form for our body, and this primarily occurs in liver, the metabolism. Sorry. Effects of metabolism vary by drug and may include the conversion from lipid soluble to water soluble compound, inactivation of drug, bioactivation of prodrug, increased or decreased toxicity. And then we have the excretion. So most drugs are filtered by the kidney. Okay, and there are various other ways of excretion also, from bile or feces, etc. So these four things are main, which is known as ADME, and this ADME is the most basics of the pharmacokinetics. So remember, pharmacokinetics is basically what our body is doing to a drug. Okay, pharmacokinetics, we understand what the drug, our body is doing to the drug. It is getting absorbed, then get distributed, then metabolized, and then excreted. So that is our body which is doing to the drug. So that is the pharmacokinetics. Okay, and pharmacodynamics, which is the other word you have heard about. So it is what the drug is doing to our body. Okay, these two are the very important terms which we need to remember in the medicinal chemistry. So I think it is clear to all what is the pharmacokinetics, and even if you have any doubt, you can just let me know in the chat box. Okay, if not, I will move toward the next problem. So moving forward to the next problem. Problem number 2 says, which of the following statements is true regarding the Lipinski's rule? Your options are: number A, log P less than 5, number B, molecular weight greater than 500 Dalton, number C, hydrogen bond donor greater than 10, and number D, hydrogen bond acceptor equal to 20. So what is the correct statement regarding to the Lipinski's rule? Again, I will give you some time, think about it and write it in the chat box, and this time I want all of you to participate and answer this question. I am giving you 5 minutes for solving this question. Then I will show you what is Lipinski's rule and we will discuss it about. Let me check the chat box now and I will discuss it. Okay, so the answers given by you is number A, number B, number B. Okay, let me check the chat box now. The statement which is true regarding to the Lipinski's rule is number A. Okay, log P less than 5. It is true regarding the Lipinski's rule. Now let us check what is the Lipinski's rule. So it is called also the Lipinski's rule of 5. Okay, and this actually relates to for the bioavailability of a drug. Okay, so the drug which follows this Lipinski's rule of 5 is more readily bioavailable for our body than the drugs which do not follow this rule. Okay, so what is this rule? So the rules are like the number of hydrogen bond donor should not be more than 5. Okay, the number of hydrogen bond acceptor should not be more than 10. The molecular weight of the drug should be less than 500, and also the C log P value or log P value should be should not greater than 5, or it should be less than 5. So this is the physicochemical properties which are necessary for a good bioavailability of a drug. Okay, so the first option is the correct one. So now let us see what is the log P. So generally P is the partition coefficient. So what is the partition coefficient? It is the measurement of a drug water solubility. Okay, partitioning means the drug will be divided into the parts between the water and oil layer. So P or partition coefficient is the concentration of a drug in the organic layer or the lipid layer, we can say here, and divided by Cw, which is the concentration of a drug in the aqueous medium or the water level. So log P is the log concentration of C in organic layer divided by concentration of drug in the aqueous layer. So log P, if it is greater than 2, then it is the lipophilic drug. Lipophilic drug means the drug is more soluble into the lipid portion or in the organic portion than the aqueous medium. And if the log P is less than 2, it is for the hydrophilic drug, means the drug is more soluble into the hydrous medium or the aqueous medium. So log P is only applied for neutral compounds. Okay, the low log P means low penetration to the CNS, and high log P means low water solubility. It's not suitable for the oral administration. So the drugs which have a very high log P, they are not suitable for the oral administration. They must be given to any other way, most suitably the intravenous way. Okay, and there are many drugs which do not follow this Lipinski's rule of 5. For example, plexitaxel it is a very famous natural product which is the which forms the drug anti-cancer drug Taxol. Okay, then we have ivermectin and we have the vincristine. So these drugs, they do not follow the Lipinski's rule. Okay, for these drugs the molecular weight is greater than 500 Dalton, but still most of the drugs, they follow the Lipinski's rule of 5 for the good bioavailability of the drug. Okay, so now moving forward, I will go to the next question. So problem number 3 says, which of the following isozymes is the most important for the drug metabolism? Your options are: number A, CYP2C9, number B, CYP2D6, number C, CYP2E1, and number D, CYP3A4. So which of the following isozymes is the most important for drug metabolism? All of you should answer this question in the chat box. Okay, even if your answer is wrong, doesn't matter, because I am not giving any marks for answering, but this type of questions will help you to understand the course better and also for your assignment purpose. So please try to answer the questions. Again, I am giving you some time. Okay, let me check the chat box now, then I can discuss it. Okay, so I think I have few answers, number A, number B. Okay, now let me check which of the following is the correct one, and then let us discuss about it. Okay, for the problem number 3, the correct answer will be number D, CYP3A4. It is the most important isozyme in case of the drug metabolism. So drug metabolism enzyme. So liver is the major organ for the drug metabolism or elimination. So there are two phases of metabolism. First is the phase 1, and then we have the phase 2 metabolism. In case of phase 1 metabolism, what happens? The oxidation or hydrolytic reactions, and in case of phase 2, what happens? The conjugative reactions happen. Sorry. So predominant enzyme system that metabolizes the drugs is the cytochrome P450 family or CYP450 family of enzymes which mediate the oxidation reactions such as the hydroxylations. Okay, so there are various isozymes for this CYP450 family. We have CYP1A2, we have CYP2C9, then we have CYP2C19, we have CYP2D6, and then we have CYP3A4. So known drugs that is metabolized by this kind of isozymes is the percentage here it is shown. Okay, for this so you can see the CYP3A4, the known drug percentage which are metabolized is the 50%. Okay. Sorry, just today my health is not too well, so sorry for the disturbance. So the role of the CYP enzymes in the hepatic drug metabolism. The relative hepatic content of CYP enzymes is the percentage, where we have this CYP3A4 for 26%, and other are the less than this, and this is the other, means there are various isozymes. Okay, and the percentage of drug that is metabolized by this enzyme, as you can see for the CYP, is the 50%, is the most one. So remember, the most important isozyme for CYP450 family is the CYP3A4. Okay. So this was the problem number 3. So now I will move forward the problem number 4. So problem number 4 says sulfation. Sulfation is an important method for detoxification of the xenobiotics. Which of the following acts as a source of a sulfate? Your options are number A, hydrogen sulfide, number B, methionine, number C, sulfuric acid, and number D, phosphoadenosine pyrophosphosulfate or PAPS. Your options are, again I will just repeat the question. It says sulfation is an important method for detoxification of xenobiotics. Which of the following acts as a source of sulfate? Your options are number A, hydrogen sulfide, number B, methionine, number C, sulfuric acid, and number D, phosphoadenosine phosphosulfate. Again I am giving you some time. Please answer the question in the chat box. A, B, C or D, which of the following options you think is the correct one? All of you please participate and write in the chat box which of the following, A, B, C or D, is the correct one. I want all of you to participate, otherwise I won't understand that you are listening to me or you are understanding the course or not. Let me check the chat box now. Okay, so yeah, the options given are number D, number D, number D. So number D is the only given answer for this question. Now let me check if the number D is the correct one. So the correct answer will be, yes, it is number D, the phosphoadenosine phosphosulfate. Okay, so this is the acts as a source of the sulfate. So this is the structure for this phosphoadenosine for phosphosulfate. Okay, then this is the synthesis of the PAPS or phosphoadenosine phosphosulfate. The abbreviation is PAPS. So this is the one-pot chemoenzymatic reaction for the A, and for B we have PAPS regeneration system coupled with chemoenzymatic synthesis of sulfated polysaccharide. Okay, so what happens generally, we have a substrate and we then get the sulfated product. So how this conversion happens? So first we have the sulfate, okay, and this sulfate is converted to the APS, which is adenosine 5-phosphate, by the the enzyme is ATPase, which converts the ATP to PPI, and then it adds one PPA or phosphate and it's mixed to PPI. And then what happens, this APS is which is adenosine 5 prime phosphate, it is converted to PAPS, which is phosphoadenosine phosphosulfate, okay, by the enzyme APSK, and the conversion, the side reaction which happens is the conversion of ATP to ADP. Okay, and this PAPS is goes to the here where we have the enzyme HS or HP sulfotransferase. Okay, this converts this PAPS to PAP. Okay, and this adds the sulfate, so which is the phosphoadenosine phosphate, and it takes the sulfate and adds to the substrate. Okay, and then we have the sulfated product. And the enzyme which regenerates this PAP to PAPS is the ASTIV enzyme, and which is the PAPS ASTI4 regeneration system. And for this conversion we have another side reaction happening, which is the conversion of the paranitrophenol sulfate to paranitrophenol, which is PNPS to PNP. So this from paranitrophenol sulfate, this enzyme is taking one sulfate and is adding up to this PAP to regenerate the PAPS, and that's how this whole enzymatic cycle happens. Okay, and this is the techniques of the sulfation. Okay, so yeah, the correct answer was number D. So now moving forward, so problem number 5, it says that alkenes and aromatic groups can be metabolized to diols. Which enzymes are involved in this particular transformation? Your options are number A cytochrome P450 enzyme, number B epoxide hydrolase, number C both of the above, and number D none of the above. So you have to identify which is the correct enzyme which are involved for the transformation of alkene and aromatic groups to diols. Your options are cytochrome P450, number A, number B epoxide hydrolase, number C both of the above, and number D none of the above. Again, I want all of you to try to answer this question. Even if your answer is wrong, it doesn't matter, I am telling you, but answering is very important. Okay, answering the questions right now, it will give you the confidence so that you can solve the questions easily during the assignments or the examination. So I want all of you to try to answer this question. I am giving you some time again. Please think and then tell me in the chat box which of the following is the correct option, A, B, C or D. Let me check the chat box now. So the answers which are given is number A, number C, number C. Okay, let me check which of the following is the correct one. So the correct answer will be number C, both of the above. So the enzyme cytochrome P450 and epoxide hydrolase, both of these enzymes are involved in the alkene and aromatic group metabolization to diols. So let us now check. So this is the enzyme epoxide hydrolase, okay, and in this epoxide hydrolase what we have a aspartate residue. So we all know aspartic acid or is a acidic amino acid, right? Means which has the free carboxylic acid group. So in most of the cases this aspartic acid, it stays as aspartate residue in sometimes, which is the carboxylate ion. So what happens, for in case of epoxide hydrolase we have this free aspartate residue, okay, and this enzyme provides the protons for the catalysis. So what happens, this aspartic acid is getting divided into aspartate and H plus ions. So this negative charge of this O minus of this carboxylate ion attacks this epoxide ring and the hydrolysis happen, okay. The first step is the attack of this carboxylate group and this formation of hydroxyl. So this is the ester intermediate is formed at first, and then after this hydrolysis of this ester, what we have this dihydrodiol product. So that's how this epoxide hydrolase, it catalyzes the metabolization of the alkene and aromatic groups to diol, and then we have this cytochrome P450 enzyme. So cytochrome P450 enzymes, they actually participates in many conversions, okay. So one important is the hydroxylation reaction. So as you can see, these are the substrate which are mentioned here, and this cytochrome P450, either it attached one hydroxyl group to the particular compounds, or it oxidizes this particular compound, okay. As you can see here, if we add some aromatic ring, we have this para position hydroxyl group in the para position addition, in the alkene also, in the normal cyclic alkene also, and then in case of the alkene we have this epoxide formation followed by this diol formation, and we have also the oxidation of this keto to this ketone group, okay. And then we have the formation of this nitrogen, addition of this hydroxyl group to nitrogen also happens in case of the amines. So that's how cytochrome P450, it catalyzes the reaction for the alkene and aromatic group for the diol formation. So remember, both of these enzymes take part into this metabolization of the alkene and aromatic groups to the diols, okay. So moving forward, in the next question I will just show you now. So problem number 6 is salicylic is a specific inhibitor of, the options are number A myo B, number B myo A, number C ACE, and number ACHE, and number D is ACE. Salicylic is a specific inhibitor of which enzyme? You have to identify. Again, take some time, but most of you are not answering, so I want all of you to now answer this question. Whatever you are thinking is the correct one, just write it on the chat box, okay. But all of you must participate answering into the live sessions, otherwise I won't understand if you are listening to me or not, or if you are understanding the course or not. Let me check the chat box now. Okay, number A, number A, number C, number A. Okay, so the most given answer is number A, and we also have number C. Okay, let us check. So the correct answer will be number A, MyoB. So selegiline is a specific inhibitor of MyoB. So what is a myo? So it is the monoamine oxidase inhibitor. Okay, so actually myo is the monoamine oxidase enzyme. So it has myo A and myo B. A and B enzyme, which is in the monoamine oxidase family. So this inhibitor selegiline is one of the MAOI, okay, which is the monoamine oxidase inhibitor. So it is a class of powerful antidepressant drugs. They are particularly effective in treating the atypical depression, panic disorder and social phobia. Due to the potentially lethal dietary and drug interactions, MyoIs have been reserved for at the last line of the treatment, and only when used when other antidepressant drugs such as SSRIs and TCAs have failed, okay, because these drugs have a very lethal dietary and drug interaction. So it is the last line of treatment for the antidepressant drug. Other antidepressant drugs are the SSRI, which is serotonin uptake inhibitors, and TCAs, okay, and then myo inhibitors or this monoamine oxidase inhibitors. So there are two types. Okay, so first is the myo A enzyme. So this enzyme is present in the cytoplasm of neurons, okay, CNS, and the peripheral tissues such as liver. It acts non-specifically on Na5-HT and dopamine, and chlorgyrine is the specific inhibitor of myo A. Okay, and then myo B is present mainly in the CNS and acts more on the dopamine. Selegiline is a specific inhibitor of myo B, and pargline is a non-selective myo I. Okay, so remember these myo A and myo B enzymes where they are present, and the inhibitors, they act as a powerful antidepressant. And also remember the examples like chlorgyrine is the specific inhibitor of myo A, whereas the selegiline is a specific inhibitor of myo B, and the paraglidine is the non-selective myo I. Okay, so now moving forward, problem number 7, it says that pK of the most acidic functional group of the antibiotic cefotaxime is 3.4. Predict where it will be absorbed in the body. Your options are: number A, stomach having the pH of 1, number B, intestine having the pH of 6, number C, in both, and number D, in none. Okay, so pK is the most acidic functional group in the antibiotic cefotaxime is 3.4. Predict where it will be absorbed in the body. Your options are: number A, in stomach where the pH is 1, number B, intestine where the pH is 6, number C, in both, number D, in none. Let me check the chat box now. Okay, so the answers are given by you is the number C, number A, number A. Okay, now let me show you what is the correct answer, and then we will discuss it. So the correct answer will be number A, stomach having the pH of 1. So how to solve this kind of problem? So the formula is the pH equal to pK plus log concentration of ionized form of a drug divided by concentration of the unionized form of a drug. Okay, so in stomach, if you will see, so now the pK is given is 3.4. So the stomach pH is the 1. So in stomach, the 1, so if you just put the value, so 1 equal to 3.4 plus log of the ionized form means the carboxylate ion divided by the carboxylic acid. So if you solve this, so you will have the value of 0.00398. Okay, so this value is less than 1, which means the concentration of the carboxylic acid, the drug will remain whole rather than the ionized form. Okay, means the concentration of carboxylic acid is greater than the carboxylate ion, means the drug is in the ionized form, is not in the ionized form, it is remain in the the drug will stay as a carboxylic acid, means it will remain unionized or as a whole rather than the ionized form. But in intestine, what happens, the pH is 6. So if you put the values again, so what we will have, the concentration of carboxylate ion divided by the concentration of carboxylic ion, means the concentration of ionized form divided by unionized form is 2.6, means the drug will be now present mostly in the ionized form. Okay, so unionized form of a drug can get a better access to the membrane, so it will be so this you have to remember, that the unionized form of a drug can get a better access to the membrane than the ionized form. In stomach, the drug will remain as a whole, that's why it will get absorbed in stomach but not in the intestine. Okay, so just remember this formula, and also remember that you have to find out which concentration is higher, the ionized form or non-ionized form, and since the unionized form is the better access to membrane, you have to find out where it gets the drugs remain as a whole, and then you have to answer this type of question. Okay, so remembering this formula and also this line, unionized form of a drug can get a better access to membrane, is very important. So now moving forward, problem number 8 says, which of the following reaction is not a phase 1 transformation, metabolic transformation? Your options are: number A, reduction of ketones; number B, conjugation to alcohols; number C, oxidation of alkyl groups; and number D, ester hydrolysis. So which of the following reaction is not a phase 1 metabolic transformation? Again I will repeat, which of the following reaction is not a phase 1 metabolic transformation? Already I have mentioned there are two phases of metabolism, okay, which happens in the liver. Okay, first is the phase 1 metabolism, and then we have phase 2 metabolism. Some reactions I have already mentioned are specifically to the phase 1 metabolism, and some reactions are the phase 2 metabolism. So now you have to find out A, B, C or D, which of the following reaction is not a phase 1 metabolic transformation. Number A, reduction of ketones; number B, conjugation to alcohols; number C, oxidation of alkyl groups; and number D, ester hydrolysis. I will give you 2 minutes. Try to find out what is the correct answer and let me know in the chat box. Let me check the chat box now. Okay, I have received few answers. Number B, number B. So most of the answers are given as number B. Let me check. So the correct answer will be yes, number B, conjugation to alcohol. It is not a phase 1 metabolic transformation, rather it is a phase 2 metabolic transformation. So this is the main your metabolic transformations. Okay, first we have a highly lipophilic drugs and we have the lipophilic drug. So if the drug is lipophilic, highly lipophilic or lipophilic, first the highly lipophilic drugs are get accumulated. So first accumulation happens, and then this accumulated, and if the drug is already lipophilic, it directly goes to the phase 1 metabolism. But if it is the highly lipophilic, first it gets accumulated and then it goes to the phase 1 metabolism, which makes this particularly lipophilic compound more polar one. Okay, and or if the drug is polar, then it goes directly to the phase 2 metabolism, and if it is hydrophilic or lipophilic, it passes through the phase 1 metabolism to make which makes it polar, and then it goes to the phase 2 metabolism, which makes the this phase 2 metabolism makes the polar drugs hydrophilic, and then this it gets excreted, and the hydrophilic drugs do not need to pass through the metabolism. They can get easily excreted after the distribution. So this is the phase 1 and phase 2 reactions. Okay, so first we have the electrophiles and we have nucleophiles. So the phase 1 reactions are the lipophilic drug which can get oxidation and hydrolysis reactions. Okay, and then we have this for phase 2 we have this glutathione conjugation reactions like acetylation, then sulfation, etc. Okay, so remember the reduction of ketones, this oxidation of alkyl group, ester hydrolysis, all are part of the phase 1 metabolic transformation, but the conjugation is not a phase 1 metabolism, it is a phase 2 metabolism. Okay, so now moving forward, problem number 9 says ibuprofen is a weak acid and it will be present in the stomach in which form? Your options are number A, ionized form mostly, number B, non-ionized form mostly, number C, half ionized, half non-ionized, and number D, it will form aggregate. This is the structure of the ibuprofen. I have mentioned this type of question just in last to last slide, I think. So now you have to identify this ibuprofen, which is a weak acid. It will present in stomach in which form? Ibuprofen, a weak acid, will be present in stomach in which form? Ionized form, non-ionized form, half ionized form, or it will form aggregate? Let me check the chat box now. Okay, I have few answers. I think number B, number B. So most of all of you are thinking it is number B. Okay, let me check. So the correct answer will be number B, non-ionized form mostly. Very good. So generally ibuprofen, it is a weak acid, okay, weak acid, so means the pK is 4.9. So it is absorbed in stomach and small intestine as a function of polarity. Charged or very polar molecule are absorbed slowly, and neutral and hydrophobic molecules absorb quickly. So the stomach, the pH is 1 or 1.5, and in the small intestine the pH is 6. Okay, so if in the just in the last slide which we have done, okay, just this one, I think, yeah, not this one. So this one, we use the formula. So if you put the formula for ibuprofen also, then you will get what is the correct answer. So that's how you have to solve it out. I haven't solved it yet, but if you put the values, you will get the same answer. Okay, so remember, ibuprofen, which is a weak acid, it's present in the stomach in mostly non-ionized form. Okay, so now problem number 10, it says which of the following statement is closest description of a phase 1 metabolism. Number A, reaction which occurs in blood supply. Number B, reaction which add a polar functional group to a drug. Number C, reactions which occur in the gut wall. And number D, all of these. So which of the following statement is the closest description of the phase 1 metabolism? Is it reactions which occur in blood supply, or reaction which add a polar functional group to a drug, or the reaction which occur in the gut wall, or all of these? I have mentioned about the phase 1 and phase 2 metabolism in last slide, I think. So I know all of you now can solve this answer very easily. Just do it quickly and let me know in the chat box which of the following is the correct answer. Which of the following statement is closest description of phase 1 metabolism? Reactions which occur in blood supply. Number B, which add a polar functional group to a drug. Number C, reaction which occur in gut wall. And number D, all of these. I will give you 2 more minutes, then I will check the chat box and I will show you the answer and we can discuss it out. Let me check the chat box now. Okay. Number B, number B, number B, number D. Okay, so number B and D are the most given answer, right? Let me check which of the following is the correct one. So the statement which is closest description of phase 1 metabolism is number B, reactions which add a polar functional group to a drug. So as I have mentioned earlier also, what happens, in the phase 1 and phase 2 metabolism. Like the phase 1 is the functionalization reactions. Okay, it converts a parent drug to a more polar metabolite by introducing or unmasking a functional group such as OH, NH2 and SH. In phase 2, conjugation reactions happen. Subsequent reaction in which a covalent linkage is formed between functional group on the parent compound or phase 1 metabolite and an endogenous substrate such as glucuronic acid, sulfate acetate or an amino acid. So metabolism or biotransformation, phase 1 and phase 2 reactions. In phase 1, reaction often make the drug sufficiently polar that can be excreted. However, phase 2 reactions are often required whereby a conjugate is added or covalently linked such as amino, glucuronic, sulfuric, acetic acid to make the drug even more polar, charged or hydrophilic. Conjugation often occurs at a functional group location. So parent drug, we have the phase 1 reaction, it converts the parent drug with the functionalization, and then the phase 2 reaction, it forms a covalent linkage or the conjugation. Although phase 1 reactions usually occur before the phase 2 reaction, sometimes this order is reversed and phase 2 reactions can occur alone. For example, the parent drug, it is from lipophilic to hydrophilic, and if it passes through the phase 1, more polar metabolite that is pharmacologically unchanged, inactive or enhanced, otherwise modified, and it gets to the conjugation, addition of conjugate, phase 2 reactions, and then it get eliminated. Okay, so number B is the correct answer, correct description or the closest description of the phase 1 metabolism. So now I will move toward the next problem. Problem number 11 says, which one of the following statement is incorrect regarding the blood-brain barrier? Your options are: number A, the walls of capillaries supplying blood to the brain are such which makes it difficult for polar drugs to leave the capillaries. Number B, the capillaries in the brain have a fatty coating making it more difficult for polar drugs to enter the brain. Number C, the inner walls of the capillaries supplying blood to the brain are hydrophobic in nature. And number D, hydrophilic drugs pass to the blood-brain barrier more easily than the hydrophobic drugs. Read all the all the options very carefully, and then let me know what is the incorrect statement or which one of the statement is incorrect regarding the blood-brain barrier. Which of the statements is incorrect regarding the blood-brain barrier? Let me check the chat box now. Okay, if you have to say anything, you can just let me know in the chat box. Okay, I am monitoring the chat box, so whoever has raised the hand, you can just let me know if there is any problem in the chat box. So I am already getting answers like number B. Okay, I will give you 1 or 2 more minutes. Just try to answer the question. Okay, let me show you what is the correct answer. So the correct answer of this question is option number D. So this statement is incorrect regarding the blood-brain barrier, which says that hydrophilic drugs pass through the blood-brain barrier more easily than hydrophobic drugs. So what happens? What is a blood-brain barrier? Let us first understand what is a blood-brain barrier. So BBB or blood-brain barrier is formed in a single layer of endothelial cells that line the inner surface of the capillaries in the brain. It is a semi-permeable capillary membrane, that is, it allows some materials to cross but prevents others from crossing. In most parts of the body, the capillaries are lined with the endothelial cells. The endothelial tissue has small spaces between each individual cell, so substance can move readily between the inside and outside of the vessel. However, in the brain, the endothelial cells fit tightly together and substances cannot pass out the bloodstream. Some molecules such as glucose transported out of the body by the special method such as active transport. Okay. So, physiology: in the brain, endothelial cells are packed much tighter. Due to this, the existence of zonula occludens, the tight junctions between them blocking the passage of the most molecule. The blood-brain barrier blocks all molecules except those that cross the cell membranes by means of lipid solubility such as oxygen, carbon dioxide, ethanol, steroid hormones, and those that are allowed by the specific transport system such as sugars and some amino acids. Substances with a molecular weight higher than 500 Daltons generally cannot cross. Okay. So, hydrophilic drugs, they cannot pass the blood-brain barrier more easily. The hydrophobic drugs can pass the blood-brain barrier more easily than the hydrophilic ones. Okay. So, the option number D was the incorrect statement regarding the blood-brain barrier. So, now next problem. Problem number 12 says low concentration of neurotransmitter dopamine has been implicated in Parkinson's disease, and L-Dopa is the most widely used medicine to slow down this disease. Which of the following statement is incorrect? Number A, dopamine cannot cross blood-brain barrier. Number B, L-Dopa acts as a prodrug. Number C, L-Dopa can cross blood-brain barrier. And number D, L-Dopa first decarboxylated by dopamine and decarboxylase before entering the brain. So, you have to find out what is the incorrect statement, which of the statement is incorrect regarding the L-Dopa, which is mostly used in the Parkinson's disease. Your options are: number A, dopamine cannot cross blood-brain barrier. Number B, L-Dopa acts as a prodrug. Number C, L-Dopa can cross blood-brain barrier. And number D, L-Dopa first decarboxylated by dopamine decarboxylase before entering the brain. Now, let me check the chat box. Okay. So, the answers are given. Number D is the most given answer. Now, let me check if the statement number D is the incorrect one or not. So, the incorrect statement is number D, L-Dopa first decarboxylated by dopamine decarboxylase before entering the brain. So, what happens generally? So, as you can see, this is the blood-brain barrier. Okay, this is the blood-brain barrier, and here we have the tyrosine which is converted to L-DOPA, and then L-DOPA can also get converted to 3-OMAD and dopamine by various enzymes. Then we have this tyrosine, which is converted to L-DOPA, and then it is converted to dopamine. Okay, and this is the dopamine transporter, which this is the post synaptic terminal of the satrium. Okay, and this is the reaction which happens generally. So we have this L-DOPA or levodopa. So what happens first, this levodopa, it is converted to this, it passes through the blood brain barrier as it is. Okay, so L-DOPA can cross the blood brain barrier as it is, and inside the blood brain barrier what happens, by using this L-DOPA is converted to the dopamine. Okay, and the enzyme which is involved here is the AADC, and then dopamine is converted to DOPAC by the MyoB enzyme. Okay, and then at the periphery, this levodopa can be converted to 3-methoxytyrosine also, and the dopamine also by the various enzymes. So problem number, the statement number D, that the L-DOPA first decarboxylated before entering the brain, is the incorrect one. So first what happens, the levodopa can cross the blood brain barrier as it is, and inside the blood brain barrier after entering, then it is converted to dopamine. Okay, and not before. That's why problem number D was the incorrect statement. Okay, now I will move toward the next problem. Problem number 13 says, which of the following equations is correct for Henderson-Hasselbalch equation for the weak base? Your options are number A, pH equal to pK minus log of unionized drug concentration divided by ionized drug concentration. Number B, pH equal to pK minus log of ionized drug concentration divided by unionized drug concentration. Number C, pH equal to pK plus log unionized drug concentration divided by ionized drug concentration, and pH equal to pK plus log of ionized drug concentration divided by unionized drug concentration. You have to identify which of the following equation is correct for the Henderson-Hasselbalch equation for the weak base. I have mentioned this equation previously when we were I was giving you the example for that mathematical equations. Let me check. I think most of you have answered it correctly. Just let me check it once. Okay, so the answers which are given is number D, number C, number C and D are the most given answer for this question. So let me check if which one of the following is the correct one. So the correct answer will be number C, pH equal to pK plus log of unionized drug concentration divided by ionized drug concentration. So this is the Henderson-Hasselbalch equation. So for weak acid, your formula is pH equal to pK plus log of conjugate base divided by weak acid, or it can be written as like for POH also it can be written that for POH, that is the conjugate acid divided by the weak base for in case of the bases. So the bioavailability in case of the pH, the Henderson-Hasselbalch equation can be used to calculate the degree of ionization of a drug. For acidic drug, the equation or the formula is the pH equal to pK log ionized by unionized. It is for the your acidic drug, but for the drugs which are weak base or basic drug, the equation is pH equal to pK plus log of unionized form divided by ionized form. So this is for the weak basic drug. So just remember, for acidic drug the formula is like this, and for the basic drug the formula is like this. Okay, and this equation you have to remember, because of for the solving the mathematical equations, the questions which I have shown you earlier also. So just remember these two equations for weak acid and weak base and the drugs, and then if you are given any kind of question with the pK and pH value, you can easily identify which is the degree of ionization of that particular drug. Okay. So now next question, which of the following is a secondary pharmacokinetic parameter? Number options are: number A, volume of distribution; number B, half life; number C, clearance; and number D, all of the above. So you have to identify which of the following is a secondary pharmacokinetic parameter: volume of distribution, half life, clearance, or all of the above. Take some time, think, and then let me know in the chat box. Now let me check the chat box. Number B, number D, number C. So I have three different answers. Okay, let me check which of the following is the correct one. So the parameter which is the secondary pharmacokinetic parameter is number B, half life. So basically when there is a drug in the systematic circulation, what happens, the drug gets distributed in the tissues, okay, and this is a reversible. So now when the distribution happens, that from the parameter which is related to this distribution is the VD, or the volume of distribution. Then we have the excretion and metabolism. The excretion forms the excreted drug and the metabolism from the metabolite. Elimination is the metabolism plus excretion, and disposition is a distribution plus elimination. So primary pharmacokinetic parameters are like clearance and volume of distribution, and secondary pharmacokinetic parameters are the half-life and bioavailability. So now let us see what is the this is the graph, which is the pharmacokinetic graph you can say. So on the y-axis we have the amount of drug in the blood, okay, and in the x-axis we have the time. So with time it gave the first the concentration of, or the amount of the blood drug in the blood, it increases, and after that it decreases slowly. So first portion up till it is increasing, it is the absorption portion, and when it is the decreasing, it is the elimination portion, and this is called the area of AUC, or area under the curve, which is the overall drug exposure. And then we have this particular concentration, which is called the Cmax. It is the highest concentration of drug in the blood. And then we have the T half, or half life, which is the time at which the drug has lost half of its maximal concentration. And then we have the C minimum, which is the lowest concentration of drug in the blood. So this graph is very important for this pharmacokinetics. In the next week, I think we will discuss more about these terms like bioavailability, half-life, all these things, and we will solve few maths also in the next class, because that is in your pharmacokinetics portion. So remember, this kind of graph is also important, where this is a this graph when first increasing the absorption increase. First portion is the absorption, where the concentration of drug in the blood is increases slowly, then it gets decreased, and the Cmax is the highest concentration of drug in the blood, Cmin is the lowest concentration, and T half is the time in which the drug has the half of its maximum concentration. Okay. So half-life is the secondary pharmacokinetic parameter, so is the bioavailability, and clearance, volume of distribution, these are the primary pharmacokinetic parameter. Okay. Now next question, problem number 15 says the process where the drug is engulfed by the cell membrane and a membrane bound vesicle carry the drug across the cell is known as number A osmosis, number B diffusion, number C pinocytosis, and number D mitosis. I will give you some time. Think, what is the process called where the drug is engulfed by the cell membrane and a membrane bound vesicle carry the drug across the cell, and what is it is known as number A osmosis, number B diffusion, number C pinocytosis, and number D mitosis. The process where the drug is engulfed by a cell membrane and a membrane bound vesicle carry the drug across the cell is known as osmosis, diffusion, pinocytosis, or mitosis. Which is the correct term for this process? Let me show you the correct. First, let me check the chat box, then I will show you what is the correct answer. Okay, so most of you have, I think, number C, number C, number C. So let me now check which of the following is the correct answer. So the correct answer will be yes, number C, pinocytosis. So generally, occasionally, polar drugs with high molecular weight can cross the cells of the gut wall without actually passing through the membrane. It involves a process which is known as pinocytosis, where the drug is engulfed by the cell membrane and a membrane bound vesicle is pinched off to carry the drug across the cell. So this is the pinocytosis. Okay, this is the extracellular fluid and the other is the substance. So what happens? The first, this substrate, this in plasma membrane gets forms a vesicle type of shape, and then it takes the substance within itself by through this vesicle. So this is another representation of pinocytosis. So these are the solutes. Solutes bind to the cell membrane, then what happens, cell membrane start to fold inward like this, and then the cell membrane continues to fold and the both ends meet at some time, and then it forms a vesicle which is known as pinosome, and then we have the nucleus, and that's how the drugs they pass into the cell. Okay, and if they do not pass the cell like this, they just form the cell membrane form a vesicle which is known as pinosome, and the process is known as the pinocytosis. Okay, so now next problem number 16 says which enzyme is involved in the activation of prodrug cyclophosphamide. Your options are number A SOD, number B cytochrome P450, number C cytochrome C oxidase, and number D MMO. Which enzyme is involved in the activation of prodrug cyclophosphamide? SOD, cytochrome P450, cytochrome C oxidase, or MMO. Which enzyme is involved in the activation of prodrug cyclophosphamide? Let me check the chat box. Okay, the options are given as number B. Okay, let me check if number B is the correct one or not, and then we will discuss the correct answer. So the correct answer is yes, number B, cytochrome P450. So this is the enzyme which is involved in the activation of prodrug cyclophosphamide. So mechanism of action, cyclophosphamide is mostly commonly used as alkylating agent. Both cyclophosphamide and ifosfamide are the first biotransformed into the hydroxylated intermediates primarily by the liver by cytochrome P450 system. The hydroxylated intermediates then undergo breakdown to form the active compound phosphoramide mustard and acrolein. Reactions of the phosphoramide mustard with DNA is considered as the cytotoxic step. So this is the initial cyclophosphamide. Then for the first step is the activation by the cytochrome P450 enzyme. After the activation, what happens? The cytochrome P450 enzyme they adds up a hydroxyl group here at this position, alpha 2 nitrogen, and then what happens? The ring opening reaction happen, and then non-enzymatic this open ring of aldehyde form a alcohol. Okay, and this is the non-enzymatic reaction, and after that this is the known as the phosphoramide mustard, which is the active form. Okay, and this after this transformation it forms a reactive species which converts to the which is actually binds with the DNA, and which is the cytotoxic step. And this not only this phosphoramide mustard is formed, acrolein is also formed, which is also toxic. So remember the first step, the enzyme which is involved for the activation of prodrug cyclophosphamide is the cytochrome P450 enzyme, where it participates in the hydroxylation reaction. Okay. So now next problem number 70 says which of the following is the correct decreasing order of drug metabolism? The options are number A liver, lungs, kidney, intestine, placenta, skin, and adrenals. Number B liver, lungs, kidneys, intestine, adrenal, placenta, skin. Number C liver, kidney, lung, intestine, placenta, adrenal, skin. And number D liver, lungs, intestine, kidneys, placenta, adrenals, and skin. Which is the correct decreasing order of drug metabolism? I will give you one more minute, then I will check the chat box and I will show you the correct answer. Let me check the chat box. Okay, so number D, number C, number B, again I have different answers for this question. Okay, let me check which of the following is the correct one and then we will discuss it. So the correct answer will be number A, liver, then lungs, then kidneys, then intestine, then placenta, then skin and then adrenals. So basically this is the pharmacokinetics and pharmacodynamics which I have mentioned also earlier. In the pharmacokinetics, what are the things that the first drug is administered, then it gets absorbed and distributed, then after that it goes to the metabolism and elimination, which is the ADME. Now what is the pharmacodynamics? The first, the drug distribution, it transfers the drug to the site of action, and then we have regulatory protein like receptor, enzyme, ion channel and transporter, which then the drug is binds to the molecular target, which it's known as its affinity, then mechanism of drug transfer, and then response of the drug, which knows as the efficacy. Then drug metabolism organs are the liver, which is the heart of the metabolism because of its relative richness of enzymes in a large amount. Schematic chart of the metabolizing organ decreasing order is the liver, lungs, then kidneys, then intestine, placenta, skin, brain, testis, muscle and spleen. Okay, and adrenals are also a part of this. After this skin this portion, the adrenals are also come here. Okay, so the correct schematic chart of the metabolism organs are this in the decreasing order. So the liver is the major or the most important metabolizing organ, then we have lungs, kidneys, intestine, placenta, skin and adrenals. Okay, so now I will move toward the next question. So problem number 18 says, which of the following statements regarding epoxide is or are incorrect? Number A, epoxides can be hydrolyzed to form diols by epoxide hydrolase. Number B, cytochromes can oxidize olefin to form epoxide. Number C, electron withdrawing aromatic rings are epoxides more quickly than electron donating one. Number D, epoxide can undergo hydride shift to form phenol. So which of the following statement regarding epoxide is or are incorrect? Your options are: number A, epoxides can be hydrolyzed to form diols by epoxide hydrolase. Number B, cytochromes can oxidize olefins to form epoxides. Number C, electron withdrawing aromatic rings are epoxidized quickly than electron donating one. And number D, epoxide can undergo hydride shift to form phenol. Which of the following statement regarding epoxide is or are incorrect? Which of the following statements regarding epoxide is or are incorrect? Let me check the chat box. Okay, number C is the most given answer. Okay, let me check if number C is the correct one. So the correct option will be very good. Number C, electron withdrawing aromatic groups are epoxide more quickly than electron donating ones. It is the incorrect statement. So cytochromes can also oxidize olefins and other unsaturated bonds to form epoxides. So now what is the reaction? So let's say olefin look like this. I will just show you. Okay, and in presence of cytochrome 450, it can undergo epoxidation to form this kind of molecule. So now the enzyme that is called the epoxide hydrolase, which open this epoxide and form the olefin, which I have shown you earlier also. So oxidation of aromatic ring can happen, and hence we have couple of possibilities. Okay, and cytochrome, just electron rich aromatic rings are likely to be epoxidized more quickly than those of the electron withdrawing substituent, and it has the consequences for the drug designing. And also there is a possible that you have a hydride shift that can occur if you can think of hydride shift which forms a phenol. Okay, so this is the epoxide or oxidant. Okay, it's also known as oxidant epoxide. The interior bond angle for is the 60 degree, which is compared to ideal angle of 109. So this is a destabilized, which has a ring strain of 13 kilocalorie per mole. So when attached to a ring, the both this C and O bonds are the cis. Okay, this cis junction is okay, but this trans junction, it do not exist because it is too much strain. Okay. So the statements, if we look at the statements again once more time, so epoxides can be hydrolyzed to form diol by epoxide hydrolase. This I have mentioned earlier in one side, that cytochromes can also oxidize the olefin to form epoxide. This also I have discussed. So just remember the electron donating aromatic groups, actually electron rich or electron donating aromatic ring are likely to be epoxide more quickly than those of electron withdrawing ones, and this can be used as a consequence for the drug design. And also epoxide can undergo hydride shift to form a phenol. Okay. So problem number 19, it says that identify the incorrect statement among the following. Number A, reduction of a flavin ring disrupts the conjugation. Number B, reduction of the coenzymes are more reduced coenzymes are more colored than the oxidized one. Number C, in FAD system, all the oxidation reduction chemistry takes place on the flavin ring. Number D, FADH2 is a reducing agent. So which of the following statement is incorrect regarding the FAD system? Number A, reduction of the flavin ring disrupts the conjugation. Number B, reduced coenzymes are more colored than the oxidized one. Number C, the FAD system, all the oxidation reduction chemistry takes place in the flavin ring. And number D, FADH2 is a reducing agent. Identify the incorrect statement among the following regarding the FAD system. I will give you 2 minutes, then I will check the answer and I will show you what is the correct answer. Let me check the chat box now. Okay, so I have few answers, number B, number C and number B. Okay, let me check. So the correct answer will be number B, reduced coenzymes are more colored than oxidized one. It is the incorrect statement. Now let us look at the FAD system. Okay, so this is FAD and this is FADH2. So after reduction of FAD ring by addition of 2 H plus and 2 electrons, we get this FADH plus, and FAD it has a flavin ring. Okay, and this in the flavin ring system, all the oxidation reaction takes place in the flavin ring only. Okay, and this FADH2, it is a reducing agent, means it can reduce the it can just reduce the other substance and get oxidized its to the FAD system. Okay, and reduction, and this reduction whenever, so as you can see we have this in the oxidized form which have this nice conjugation. Okay, and whenever there is this kind of conjugation, the compounds are gets generally they get very bright colors. Okay, but in the reduced enzymes, what happen, if you can see this conjugation is now broken, so now that's why this reduced enzyme coenzymes are less colored than the oxidized one. Okay, and the reduction of flavin ring, it disrupts the conjugation, and this is also the correct statement. So as you can see, from FAD to FADH2, the flavin reductase enzyme is works, and it converts NADH to NAD plus. Okay, and this is the other transformation which can take place into the flavin ring. So remember, reduction of the flavin ring disrupts the conjugation. The reduced coenzymes are less colored than the oxidized one. In FAD system, all oxidation reduction chemistry takes place in the flavin ring, and FADH2 is a reducing system. So now last question of today's session, it says which of the following reaction is not catalyzed by flavin dependent enzymes. Your options are: number A, alcohol to ketone conversion; number B, formation of disulfide bond; number C, oxidation of phenol to catechol; and number D, carboxylic acid to aldehyde formation. Which of the following reaction is not catalyzed by flavin dependent enzymes? Alcohol to ketone, formation of disulfide bond, oxidation of phenol to catechol, and carboxylic acid to aldehyde conversion. Which of the following reaction is not catalyzed by flavin dependent enzymes? Let me check the chat box. Okay, so I think I have multiple answers: number C, number B, number C. Okay, and now let me check which of the following is the correct one, and then we will discuss it, and it is the last question of today's session. So the correct answer will be number D, carboxylic acid to aldehyde. So this reaction is not catalyzed by the flavin dependent enzyme. So generally flavin dependent enzyme, they participate into the your oxidation kind of reactions. Okay, like alcohol to ketone conversion, oxidation of phenol to catechol. Carboxylic acid to aldehyde reaction, it is the reduction kind of reaction. Okay, and formation of disulfide bond and this kind of conjugation reaction also is catalyzed by the flavin dependent enzymes. So not the most flavin dependent enzyme bind the flavin in a non-covalent manner. Some of the enzymes actually bind to the covalent manner. So this covalency bond happens at the 8th alpha position or the 6th position. So this is the flavin ring. Okay, and this we have this 8 alpha position, and then we also have a 6th position. Okay, and this one. So we will not go to many details, but let us say the example. So highly conjugated isoalloxane tricyclic ring system in the flavins is an excellent electron acceptor and this is responsible for the strong redox properties. Flavins can accept either 1 electrons at a time or 2 electrons simultaneously in overall 2 electron oxidations. It is not clear if the reaction processed by single 2 electron transformation or 2 electron transformation steps. So once the flavin ring is reduced, now the enzyme requires second substrate to return the flavin to oxidized form, which can happen from the substrate molecules. So some enzymes are called oxidases and other are called dehydrogenases. Okay, so basically flavin enzymes can act as the oxidases or the dehydrogenases, and the reactions are mostly the oxidation kind of reaction or the hydroxylation kind of reaction, which are catalyzed by the flavin dependent enzyme, not the reduction kind of enzymes. Okay, so the number D was the reaction, the conversion of carboxylic acid to aldehyde. It is not catalyzed by the flavin dependent enzymes. So this was the last question of today's session. So today what we have discussed the pharmacokinetics, the absorption, admittance, and then we discussed mostly about the metabolism, the several metabolic enzymes and organs. So I will share you my email ID in the chat box. So this is my email ID. If you have any doubt or any query regarding today's session or any other previous sessions, you can just drop an email to me in this ID. Okay, I will try to answer your query. And today we had our week 5 live session, and we discussed the week 6 sessions. This kind of problem solving sessions we are going to have every week till your course is finishing, and after at the end we will have also summary session to summarize the points which I could not discuss during the class due to the time foundations. Okay, and from next Tuesday, what we will discuss the week 7 course content in our next class. So I want all of you to join on time. Okay, this kind of classes we are going to have every Tuesday from 6 p.m. to 8 p.m. and this is the joining ID, which is same for each and every class. So this is my email ID. Now again I am saying, if any problem arises, just let me know through the email, and also you can just drop an query in the discussion forum of SWAYAM portal also. Okay, this lecture PPT and the video of today's class I will upload it soon, so you can also these two things you will find it on the SWAYAM portal at the portion which is called the problem solving session. If you go to the problem solving session portion, you will find this all lecture PPTs and videos there. Okay, so now I will close the session for today. I thank you all for joining today's class, and I hope you have understood the week 6 course content a little better, and now you can solve the assignment questions more easily. So if you have any doubt, any query, please let me know. Otherwise I will just close the session for today. And anyways, you can just reach me through the email also. The email ID is given here, so just feel free to contact me if any problem or any doubt arises. I will close the session for today. So I thank you all for joining. We will meet in the next Tuesday from this time 6 to 8 p.m. So thank you all. I am closing the session for today.